So I'll try to not make it go the whole time.
Are you going to lecture at all on Wednesday too?
No, because on Wednesday we have 3 labs to discuss because you have a lab exam next week. So that's going to take us time, and I don't want to also do that. We're probably not going to get to 2 topics, I would guess. Yeah.
Quite a few chapters.
Yeah, we'll get through mutation and DNA repair and gene regulation. We'll go really quick. And then we'll get to simple and complex patterns of inheritance and probably population genetics. And then, well, actually, it's 3 topics. Animal development, immunology, and biotechnology. But what I'll do is I'll post recorded lectures for those. They won't be on the final, but in terms of getting that information, I'll post recorded lectures for all those topics. And obviously, if you guys have any questions, just email me over the break or into next semester, because you guys have access to that till I think the 5th week of next semester. The most into, I think, there's nothing that you're going to need in order to take 1114. But in terms of future bio classes, I would definitely watch the animal development and the immunology. The biotechnology is useful, but you'll probably learn it in other classes. So, okay. Okay, so let's talk about gene expression. Just to remind you about a couple of things that we talked about in the last class. So what we're talking about in gene expression is how do you go from a segment of DNA that codes for a protein, a segment of DNA called a gene that codes for a protein to actually producing that protein. And you have to go through the process of transcription and translation. And then right after we discussed that, we just compared gene expression in a bacterial cell, which you had transcription and translation happening in the same area, which is a cytoplasm or cytosol. And then we talked about and compared that to gene expression in a eukaryotic cell. And we said that transcription happens in the nucleus. Then you also have what's called RNA processing. That also happens in the nucleus. Then you have export of the RNA out of the nucleus. And then you have translation happening in the cytosol or cytoplasm. So they're spatially separated. And what we went through in the last class was we went through transcription, and that applied to both eukaryotic and bacterial cells. And then we had just started to talk about RNA processing, which is typically, which happens typically in eukaryotic cells and not typically in bacterial cells. And again, this is going to happen in the nucleus of a eukaryotic cell. And this is comparing, obviously, a eukaryotic cell to a prokaryotic cell in terms of gene expression, but it's also highlighting something else that we haven't talked about yet. I think I mentioned it last week. So when you look inside the cells here, this is supposed to represent a typical eukaryotic gene, and this is supposed to represent a typical bacterial gene. And when they have it like that, the dark orange segments are supposed to be the regions that code for the protein, and then the light orange segments are supposed to be regions that do not code for the protein. Okay, so what you can see here is that in a bacterial cell, the gene is one long coding region. Okay, so typically the gene is one long coding region. So you can transcribe that, and at the same time that you're transcribing and making the RNA, you can actually start to translate it and make the protein. What happens, in a typical eukaryotic gene, your coding regions are interrupted by these non-coding regions. And then we give those 2 types of segments within a gene 2 names. So the coding regions are called exons. So exons are the regions that code for protein. And the regions that interrupt, the non-coding regions that interrupt, are called introns. Okay? And then what's going to happen in eukaryotic cells is when you transcribe this gene, you actually transcribe both the exons and the introns. So you initially produce this RNA molecule that contains both. So the RNA molecule that you initially produce contains the introns and the exons. And then you have to process that initial RNA molecule you produce before you can export it out of the nucleus and into the cytoplasm. Okay, so RNA processing, and specifically we're talking about mRNA processing, is going to start with what we'll call pre-mRNA. In the slide there, they're calling it the primary transcript. But you're going to start with pre-mRNA, and then the product of mRNA processing will be mature mRNA, and this is the type of RNA that can be translated. And there's several things that have to happen during RNA processing, and they're up there on the board, but I'm going to write them just slightly differently. You have to have the addition of a 5' cap, and we'll talk about what that is. You have to have splicing, and splicing is the process where you take out those introns and you attach the exons together. Okay, so during splicing, you're going to take out the introns that are in that pre-mRNA molecule, and you're going to paste together or attach together the exons. And then you also get addition of what's called a poly-A tail, and we'll talk about what that is. And that gets added to the 3' end of the mRNA molecule. Okay, so basically you add things onto each end of the mRNA molecule, and then you also have splicing, and that's RNA processing. And then that's going to produce a mature mRNA molecule. And again, all three of those things are happening in the nucleus. And then once you produce the mature mRNA, then it can be exported out of the nucleus and into the cytoplasm where it can be translated. Okay, so let's talk about the 5' cap and the poly-A tail. So first we're going to describe what they are. So at the 5' end you add, we'll just call it a modified guanine nucleotide. Okay, so modified guanine nucleotide. That gets added to the 5' end, and that modified guanine nucleotide is what the 5' cap is. Okay, so the 5' cap is just a modified guanine nucleotide that gets added to the 5' end. And then to the 3' end, you get the addition of this poly-A tail. And the poly-A tail just consists of a bunch of adenine nucleotides attached onto the end. So what happens is this enzyme comes in and is going to add a bunch of adenine nucleotides on to the 3' end, and all those adenine nucleotides are referred to as the poly-A tail. And again, that gets added to the 3' end. Okay, in terms of why they're functionally important, there's only one that I want you to remember, but I'll mention two others. So the one that you need to remember is that the 5' cap and the poly-A tail are both important in helping ribosomes bind to the 5' end during translation. Okay, so remember that after you process this RNA molecule, it's going to go out into the cytoplasm. Ribosomes are going to translate it to make the protein, and the 5' cap and poly-A tail are important for the ribosomes being able to bind to the RNA molecule at the 5' end. Okay, in addition to that, they both help to prevent degradation of the mRNA, so breaking it down. And then they also both seem to facilitate the export of the RNA out of the nucleus and into the cytoplasm. So those last two you don't need to remember, but those are also additional functions of the 5' cap and poly-A tail. Okay, and then splicing. Again, what you're going to do during splicing is, here you can see the pre-mRNA, and the exons are yellow and the introns are green. And splicing is when you take out these green segments, which are the introns, and then you attach the exons together. So in splicing, you're going to take out the introns and paste together the exons. And the complex, I thought I had it up here. The complex that performs that process is called a spliceosome. So this is the name of the complex that actually performs the splicing. It's called a spliceosome. And it's actually a complex that's made out of proteins and these RNA molecules called snRNAs. You don't need to know that for this class. but these complexes include molecules of SN, RNA, and proteins. And so it's multiple proteins, multiple snRNA molecules come together to form a spliceosome. And when you're looking at this picture, all of this right here, the blue subunits and then this green subunit, that's all part of one spliceosome. And that's going to cut out this intron and paste together these two exons. Okay. And then that's the last thing in terms of RNA processing that you need to know. There are also another thing that happens in terms of splicing is alternative splicing. And alternative splicing is splicing the mRNA in different ways. Okay. So what they're showing you here is this is the pre-mRNA with the exons and the introns being in yellow. And then they're showing you 3 different ways, and there's more than 3, that you could alternatively splice this mRNA molecule. So you could take exon 1, attach it to 2, and then leave 3 out, attach 2 to 4, 5 to 4, and then 6 to 5. And alternatively, you could have a splicing where you do exon 1 to exon 2 to exon 3 and include all the exons. But then there are alternative ways of splicing where you also not only cut out the introns, but some of the exons in the splicing process. And the advantage of this is that this is a way that a single gene could code for multiple proteins, because when you splice it differently, that's going to end up in a different protein being produced. Okay? So a different protein is going to be produced if you splice the mRNA molecule differently. So that's a way that one gene could code for multiple proteins. Okay? And then this is just this is something you don't need to remember for an exam or quiz. But when you're looking at this gene here, the alpha-tropomyosin gene, I took this picture from a different textbook. And this is showing you alternative ways of splicing it. And then this is showing you specifically what cell type that form of splicing takes place in. So meaning that the way it's spliced depends on the cell type that you're in. And that would give rise to a different protein in those different cell types. And that protein would be playing a slightly different function in each of those cell types. So the way of splicing a gene can be cell type specific depending on the gene that you're talking about. Okay. So that's RNA processing. Do you guys have any questions about RNA processing? Okay, so the next process we're going to talk about is translation. And this is where you're going to take an mRNA molecule, and that's going to be the template, and you're going to use that to synthesize a protein. Now, if you remember from transcription, you were using a segment of DNA to make a molecule of RNA, and in terms of acting as a template, you just had to use your base pairing rules in order to determine the sequence of nucleotides. But in translation, you're going from a code of nucleotides, which is the mRNA molecule, into a code of proteins. And so you're going to have to use something to translate that message, okay? And you use, the cell obviously isn't reading this in the cell, but when you're asked to translate mRNA sequences on exams and quiz, well, just exams now, you're going to have to use the genetic code. You don't need to memorize it, you just need to know how to use it, okay? And the other key thing, one is obviously knowing how to use it, and the other key thing is to realize that you always read the mRNA sequence. Okay, so when you did the lab last week, a common mistake that people make during that lab is they read the tRNA sequence, they look up the tRNA sequence in the genetic code, and you're not supposed to do that. You always look up the mRNA sequence. Okay, so you're going to be reading the mRNA. And what's going to happen is you're going to be given an mRNA sequence, and I won't break it up into 3. I'll explain why I'm doing that in just a second. So let's say this is your initial mRNA sequence. So when you're using the genetic code is, you can think about it, it's broken up into 3-letter words that are called codons. Okay, so each of these 3 letter, you can think of them as words, is a codon, and each codon specifies a particular amino acid. Okay, so when you look at the mRNA sequence, and you always start with we'll get to this, you're going to start with the start codon, which is always AUG. You want to look up AUG in the genetic code. So when you look at the genetic code, the letter in the first position will tell you what row to go to, so that's why it says first position here. So because the first letter is an A, you want to come down to this row. And then you need to find what letter is in the second position. Oh, it's a U. So I'm looking for where A and U crisscross. And then you can see that AUG is right here, and that is the start codon, but the start codon also codes for methionine. And so when you're building your protein, you're going to start with methionine. Okay? And then you're going to read after the start codon. The start codon isn't directly at the beginning of the mRNA molecule like I'm drawing it here. There's actually some untranslated mRNA that comes before it. So when you're translating, you're going to start at the start codon, and then from the start codon, you just read in 3 nucleotide bursts. So then you just read the next 3 nucleotides after that. That's the next codon. So now you're looking for AGC in the genetic code. A tells you to come down to this row again. G is in the second position, so you're coming all the way over to this column. And then when you look within this group here, here's AGC, and that codes for serine. Okay, so then after you add the methionine in, you're going to attach serine to that methionine. And then you're just going to read in 3-letter blocks as you move down the mRNA molecule. Do you guys have any questions? Yeah.
Does it always start with AUG?
Yeah. Other questions? Okay. Also, another thing to realize about the genetic code is that our what we call it is degenerate. So we say the genetic code is degenerate. And what that means is that more than one codon can specify the same amino acid. Okay, so when you hear the term that the genetic code is degenerate, that means that more than one codon can specify the same amino acid. And just to give you an example of that, you can see that UUA, UUG, and then also these 4 codons here all code for leucine. So if the ribosome encounters any of those 3-letter words, it's going to add a leucine in to the protein that's making. So the fact that more than one codon can code for the same amino acid means that the genetic code is degenerate. Okay, so any questions on how to use the genetic code? Because you will have to use it on the final. There will be a question where you have to translate. Okay, so let's talk about the different components that you need for translation, and then we'll talk about how the components are used talk about how the components interact with each other to produce a protein. So the first thing that you're going to need for translation is a template, and that's going to be mRNA. Okay, so if I ask you what is mRNA's role in translation, you're going to say that it's the template. Okay, so mRNA's role is that it's the template. And I told you that in mRNA you have these 3-letter words called codons. So you should know what a codon is. And so a codon is a 3-nucleotide long sequence. Okay, so a codon is a 3-nucleotide long sequence that codes for an amino acid, or sorry, let me add something in there. 3-nucleotide long sequence found in mRNA. So it's a 3-nucleotide long sequence found in mRNA that codes for an amino acid or stop. Okay, so 3-nucleotide long sequence found in mRNA that codes for an amino acid or stop. So just to go back to the genetic code really quickly, there's 3 codons that do not code for an amino acid but code for stop. So unlike the start codon, which also codes for an amino acid, the stop codons do not code for an amino acid. It just tells the ribosome that you're supposed to stop making the protein at that point. Okay, so in an mRNA, you're going to have, an mRNA molecule will have multiple codons, and it depends on how big the protein is you're trying to make. So the larger your protein, the more codons you're going to need because the more amino acids you need to code for. And then in addition to that, every mRNA molecule will have a start codon. And you can see here now that the start codon isn't at the very beginning of the mRNA molecule. It's kind of sitting within it. And the start codon signals the beginning of translation, but this is also another key point. So it signals the beginning, but it also sets what we call the reading frame. Okay, so it's going to signal the beginning of translation, but it also sets what we call the reading frame. And what I mean by that is that it sets or determines the three-letter words that you're going to read as you move down the mRNA molecule. Okay, so the start codon determines the three-letter words that you're going to read as you move down the mRNA molecule. So basically what's going to happen is, again, the AUG is not at the beginning of the mRNA molecule, but the ribosome knows to start here because this is the start codon. And then once you say that you're going to start here at the start codon, the ribosome just reads the next three nucleotides after the start codon. Then it reads the next three nucleotides after that. So the start codon determines what those three-letter words are going to be after it. And that's the reading frame. And the start codon sets that. Okay, and then so you have a start, so in an mRNA molecule you're going to have a start codon, multiple codons, and then a stop codon, and that's going to signal or tell you where to stop. Okay, so the stop codon will tell you where to stop. Okay. Do you guys have any questions about codons or the start or stop codon?
So the question that we'll have on the exam, like we'll be given the table, are we just going to be writing down what amino acid it codes for?
What's that?
You said that the genetic code.
Oh, well, okay, so one thing about the final, I didn't tell you guys, is that one, there won't be a part two, it'll be all multiple choice. So you won't have to write anything, well, you'll have to fill in circles. Yeah, so basically the type of question that you're going to see is you'll be given a sequence of mRNA and have to determine the amino acid sequence from that out of multiple choices. So yeah, you'll have to take an mRNA sequence and translate it using the genetic code and write out the amino acids. But then you're obviously looking within a multiple choice question which one matches your amino acid sequence. Is that what? Correct. You do not have to memorize it, yeah.
So what if, like, due to some sort of transcription error, like we have AC, UG, what if that C was swapped out?
Where?
For an amine, the second line.
This one?
Yeah. No, from the very left.
Oh, here?
Yeah. Then it would be AUG. Is there some sort of way that the cell has?
Oh, I see what
I'm
you're saying.
saying it would start there and then the whole thing would be.
It depends on what organism you're in. There's a special sequence that sits before the start codon in bacteria that tells the ribosome that that's where it's supposed to bind, and it's right next to the AUG. So I would assume, this is what I'm assuming in bacteria, that if it was AUG here, it wouldn't because it doesn't have that previous sequence to help the ribosome bind there. Now, in eukaryotic cells, I don't know the answer, but if I remember correctly, what's going to happen in well, no, I know this part. The ribosome is going to bind to the 5' end, and then basically it scans the mRNA looking for the start codon, and I believe it goes to the first start codon that it finds, but I'm not positive about that. So in that case, if that's what happens, then that certainly would affect where you started, yeah. And what we're going to do is after this, we talk about different types of mutations. So not necessarily mistakes in transcription, but we're going to look at mutations and how they can mess up translation, and then what that results in terms of what types of proteins you produce. Because you can also change a normal codon into a stop codon, and that's going to cause you to stop prematurely, and then you end up producing the shortened protein. Okay, so then that's mRNA, and I'm going to just point out that next we're going to talk about tRNA, and one important part of the tRNA you need to know is an anticodon, okay? So an anticodon is a 3 nucleotide long sequence found in tRNA. Okay, so an anticodon is a 3 nucleotide long sequence found in tRNA that is complementary to one of the codons. Okay, did everybody got that, or you want me to repeat it? Okay, and just you should also note that a tRNA molecule is only going to have one anticodon. So I showed you mRNA is going to have multiple codons, but each tRNA molecule only has one anticodon that's complementary to one specific codon. Okay, and by complementary, what I mean is they're showing you this is one tRNA molecule. They would not be connected like this, so ignore this bottom part of the picture, but this is the anticodon for this tRNA molecule. And UAC is complementary to AUG because U pairs with A pairs with U, and C pairs with G. So that's what I mean by the anticodon being complementary to one of the codons. Okay, is everyone okay with that? Okay, so this is what a tRNA molecule looks like. And so in this picture, the anticodon is GGC, and you can see that it's at this end of the tRNA molecule. And then what's not shown in the picture is that there'd be an amino acid attached to this end. Okay, and it's specific. Each tRNA molecule with a specific anticodon has a specific amino acid attached to it. Okay, and then in terms of the role of tRNA during translation is that tRNA is what brings the amino acids in to be added into the growing protein that you're making. Okay, so each tRNA molecule is going to carry in an individual amino acid to be added in to the growing protein during translation. Okay, so I told you that each tRNA molecule is going to have a specific amino acid attached to it. So this anticodon is GGC. That anticodon is complementary to what codon?
CCG.
Right, CCG. So when CCG, so let me just draw this, the lines being part of a larger mRNA molecule. So whenever you come up with CCG, that means that tRNA molecule is going to come in and bind because its anticodon is complementary to that codon. And then this would be part of a larger tRNA molecule. And then at the other end, you have an amino acid attached to that tRNA molecule. And so when this comes up, it carries in this amino acid. Okay, so what amino acid is going to be attached to this tRNA molecule? So the tRNA molecule that has an anticodon GGC, what amino acid is going to be attached to that tRNA molecule? Sorry, I haven't had a chance to look it up yet. Right, no, proline. Whoever said proline's right. So one thing that's tricky is that you're looking up CCG. Because remember, when you read the genetic code, you're always reading the mRNA molecule. But another way to look at what we just did is that the reason that CCG codes for proline is because there's a tRNA molecule in the cell that has an anticodon complementary to CCG, and then at the other end of that tRNA molecule you have proline attached to it. So that's the reason that CCG codes for proline. Okay, do you guys have any questions about tRNA molecules? Okay, there's also a class of enzymes that specifically attach an amino acid to a tRNA molecule, and those are called aminoacyl tRNA synthetases. Okay, so aminoacyl tRNA synthetases, they're enzymes that are going to attach an amino acid to a tRNA molecule, and a tRNA molecule with an amino acid attached to it is called an aminoacyl tRNA. That's why they're called aminoacyl tRNA synthetases. because they synthesize aminoacyl tRNAs, which is a tRNA molecule with the amino acid attached to it. Also, another name for an aminoacyl tRNA is a charged tRNA. So if you hear the word or phrase, it's a charged tRNA molecule, that means that it's a tRNA molecule that has had the amino acid attached to it. Okay. The next thing that you're going to need in translation are ribosomes. And you already know these as the complexes that synthesize proteins inside the cell. And so their role in translation is that they're the enzymes that connect together amino acids to synthesize the protein. Okay, so the ribosomes are the enzymes that connect amino acids together to form the protein. Additionally, they're going to be the site that brings together the mRNA molecule with the multiple tRNA molecules that need to be used during translation. Okay, this is showing you bacterial chromosome and a eukaryotic chromosome, and they're very similar to each other. They both have a large subunit. And a small subunit. Okay, so every ribosome is going to have a large subunit and a small subunit. And both subunits are made out of one or more RNA molecules and multiple proteins. Okay, so the ribosome as a whole is a complex of multiple rRNA molecules. Remember, R stands for ribosomal RNA, and then multiple proteins. And all they're doing in this picture, you don't need to know the specific. You need to know that they have a large and small subunit, but you don't need to know there's a detail down here. It's showing you what RNA molecules make up each subunit and then how many proteins make up that subunit. That detail you don't need to know. But this is just showing you a bacterial ribosome, and this 30S subunit is the small subunit, and the 50S is the large. And the same thing over for the eukaryotic. Here's the large, and here's the small. Okay. And when they're not translating an mRNA molecule, the two subunits are separate. Okay, so they're only attached to each other when they're translating. Okay, there's also three important binding sites for tRNA that you need to know for a ribosome. One is called the amino acyl site, and in the picture that's the A site. Another is called the peptidyl site. And that's the P site. And then you also have an exit site. That's probably the easiest to remember. That's the E site. Okay, so you have an amino acyl site, a peptidyl site, and an exit site. And these three sites within the ribosome are going to have tRNA at some point bound to them. Okay, so I'm going to state it, but also use this animation to show you what each of the sites binds. So this is showing you a ribosome that is in purple there. Sorry, this is the small subunit of the ribosome in purple, and this is the large subunit of the ribosome. And then it has three sites, an A site, a P site, and an E site. And what we'll learn is that this is the start codon here. And during the initiation stage, what happens is that you're gonna, everything's going to become complex together such that this first tRNA molecule sits in the P site. Okay, that just ignore for right now. And so what the A site binds, the A site binds the tRNA molecule carrying the next amino acid to be added into the growing protein. Okay, so the tRNA carrying the next amino acid to be growing in the protein binds to the A site. So what you're going to see in the animation here is a tRNA molecule right there carrying proline, and proline's the next amino acid, and that tRNA molecule comes in and binds to the A site. So all the tRNA molecules that are coming in will first bind to the A site. Okay, so that binds the tRNA carrying the next amino acid to be added into the protein. And then what's going to happen during translation, we'll show you the first step here, is you're going to take the methionine off this tRNA and attach it to proline. Okay, and when you do that, now you have this tRNA molecule that carries proline and then methionine. Okay, and then you're going to move down 3 nucleotides to the next codon. And what's going to happen is when you do that, that obviously moves this tRNA molecule now to the P site. And the P site always binds the tRNA molecule carrying the growing polypeptide chain. So right now your growing polypeptide chain only carries methionine and proline, and the tRNA molecule carrying that growing chain is what the P site binds. And then you can see that the E site is where the empty tRNA molecules are going to leave. Okay, so the E site is where they leave. So if you do this one more round, you can see that the next tRNA molecule carrying the tRNA molecule carrying the next amino acid to be growing into the polypeptide chain comes in now and binds to the A site. Then what's going to happen is you're going to take proline off and attach it to tyrosine. So then you do that, and you can see you have the shift occurring at the same time. And now the tRNA molecule carrying the tRNA molecule carrying the growing polypeptide chain is again in the P site. This tRNA molecule is going to leave through the E site, and your next tRNA molecule is going to come in and bind to the A site. Okay, do you guys have any questions about the 3 different sites for tRNA and ribosomes? Okay, so what we're going to do next is watch this animation that describes translation, and then we'll talk about each of the steps of translation on the other side.
In prokaryotic cells, translation is initiated by formation of an initiation complex consisting of the 30S ribosomal subunit, formal methionine tRNA, and messenger RNA. The 50S ribosomal subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown. The 70S ribosome has 2 sites to which transfer RNA-carrying amino acids can bind. One is called the peptidyl, or P site, and the other is called the acceptor, or A site. There is also a third site called the exit, or E site, where transfer RNAs are released. The initiating transfer RNA carrying formal methionine binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site. The formal methionine carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site by a peptide bond. The ribosome now advances a distance of 1 codon, and the transfer RNA that carried the formal methionine is released at the E site. A transfer RNA carrying the next amino acid now moves into the A site, where the anticodon on the transfer RNA matches the codon on the messenger RNA. The ribosome shifts down by a distance of 1 codon. As the shift occurs, the 2 amino acids on the transfer RNA in the P site are transferred to the new amino acid, and the second transfer RNA is released from the E site. The ribosome continues to move along the messenger RNA, and new amino acids are added to the growing polypeptide chain. Elongation of the polypeptide is terminated when a stop codon moves into the A site. A stop codon does not specify an amino acid and does not have a corresponding transfer RNA. The ribosome dissociates into the 30S and 50S subunits, and the messenger RNA and protein are released.
Okay, so this process is a little bit more complicated than transcription, but we're going to break it down into the same 3 stages that we broke transcription down into. So we have an initiation stage, elongation stage, and the termination stage. And so first we'll go through what generally happens in each of these stages. Oh, that's the red one that works. And so we'll start with the initiation stage. And so the first thing that happens, well, pretty much the only thing that happens in the initiation stage is the mRNA, the tRNA bearing the first amino acid, which we call the initiator tRNA. So the mRNA, the tRNA bearing the first amino acid, and the two subunits of the ribosome come together. Okay, so that all happens in the initiation stage. The mRNA, tRNA bearing the first amino acid, and the two subunits of the ribosome are all going to come together and form a complex during the initiation stage. And then the next stage is the elongation stage. And during this stage, the ribosome reads the mRNA molecule from the 5' end to the 3' end. Oops, not ribose. So it's going to start at the 5' end and then read towards the 3' end, but reads in parentheses. And synthesizes so the ribosome reads the mRNA 5' to 3' and synthesizes the protein from the N terminus to the C terminus. Okay, so the ribosome reads the mRNA 5' to 3' and synthesizes the protein. From the N terminus to the C terminus. And then the termination stage is the last stage. And during this stage, the ribosome reaches a stop codon. Protein is released. And everything disassembles, meaning the large and small subunit come apart and release the mRNA molecule. Okay, so this is essentially, in general, what's going to happen regardless of whether you're in a prokaryotic or eukaryotic cell. These are the different things that are going to have to happen during the initiation, during the elongation, and then the termination stage. Okay, so now we're just going to discuss some specifics of each of the stages, and in some of the specifics we're going to compare and contrast what happens in prokaryotic cells or bacteria versus what happens in eukaryotic cells. So the first thing we're going to talk about is the initiation stage. And again, one thing I told you what I told you has to happen is you have to have the mRNA, tRNA bearing the first amino acid, which is called the initiator tRNA, and the two subunits of the ribosome come together. And this is going to happen slightly differently in bacteria when you compare it to eukaryotic cells. And so In bacteria, the small subunit of the ribosome can either bind the mRNA first or it can bind the initiator tRNA first. So you can just remember it as it's drawn in your textbook, so just think about it as binding the mRNA first, but it can bind either of those in either order. But what happens in bacteria is the small subunit of the ribosome recognizes this short nucleotide sequence that's just upstream or comes just before the start codon. So that's how the ribosome knows to bind here, because there's this short sequence here that tells it that's where it's supposed to bind. So then that brings in the small subunit, and then the initiator tRNA comes in and binds because its anticodon is complementary to AUG. And then the large subunit will come in and complete the complex, and then at the end of the stage what you have is everything together and the initiator tRNA sitting in the P site. Okay, and then that's going to kick off then the elongation stage. Okay, in eukaryotic cells it's slightly differently, happens slightly differently. What happens? The end result is the same. In eukaryotic cells, the small ribosomal subunit binds the initiator tRNA first, and then that complex binds to the 5' cap. Okay, so the small subunit of the ribosome with the initiator tRNA already bound to it binds to the 5' cap, and then what happens is the large subunit will come in. Before that, I skipped a step. So once it binds to the 5' cap, then it's going to scan the mRNA looking for the start codon. So the small subunit with the initiator tRNA bound to it is going to start at the 5' cap and scan looking for the start codon. Once it finds it, then the large subunit comes in and completes the complex. And again, at the end of the stage, the initiator tRNA is sitting in the P site. Okay, and then once that's complete, then the elongation stage is going to start, and it's going to start with the tRNA carrying the next amino acid coming in and binding to the A site. Okay, so then that's going to enter the elongation stage then. And so basically, this is showing you the cycle that happens during the elongation stage. And this cycle is going to keep on going until you bring a stop codon into the A site. Okay? So what you can see here is that you've already produced some of the protein. So in this picture, these teal molecules are the tRNA molecules, and the purple shapes are supposed to represent amino acids. And so right now, you have this codon sitting in the A site, and that codon that's sitting in the A site determines what tRNA molecule is going to come in and bind there, because it has to be a tRNA molecule with an anticodon complementary to that codon. So then this tRNA molecule comes in and binds to the A site, and then what you're going to do is you're going to take this growing polypeptide chain, which is bound to this tRNA molecule, and you're going to attach it to this amino acid. Okay, then after that step, you're going to have an empty tRNA molecule, and then you have a tRNA molecule carrying the growing polypeptide chain. And then there's a series of movements that moves everything down by 3 nucleotides. So that brings the next codon into the A site. It moves this tRNA molecule to the P site, so that now it's bound to, the tRNA carrying the growing polypeptide chain is bound to the P site, and then that empty tRNA molecule leads to the E site. And then basically, now you've brought the new codon into the A site, and you go through this cycle again, and you're going to keep on going through this cycle, moving down 3 letters at a time until you bring a stop codon into this A site. Okay, so you're going to continue with that cycle, and then once a stop codon gets brought into the A site, there's not a tRNA molecule for the stop codon. Okay, so there's no tRNA molecule for the stop codon. Also remember, the stop codon does not code for an amino acid. What happens is when the stop codon gets brought into the A site, it's actually a protein called release factor. And this protein release factor comes in and binds to the A site, and that initiates the release of the protein, and then the small and large subunit separating from each other and releasing the mRNA molecule. So it leads to everything disassembling and the release of the protein. Okay, do you guys have any questions about some of the specifics that we talked about in terms of the different stages? Okay, just two additional things. It's mostly vocabulary. The first thing is, what is a polyribosome? So typically what you have in a cell is you're going to have multiple ribosomes bound to the mRNA molecule and translating it. And they would just be at different points in translating the mRNA molecule. And the mRNA molecule here with all these ribosomes bound to it, that's what a polyribosome is. So it's just an mRNA molecule with multiple ribosomes bound to it and translating it. And again, they would just be at different points in reading that mRNA molecule and producing that different, what do you call it? A different time point in reading the mRNA molecule and producing that protein.
And they're all reading the entire thing and just producing copies of that protein.
Right, so every ribosome is going to start here at the 5' end, bind, and move towards the 3' end. So every ribosome here started here and will end here. This one bound first, then this one, and then this is the last one to bind of the one shown in the picture. And they're all going to end up producing the same protein, too, yeah. So if it binds the same mRNA molecule, it translates it in the same way and will produce the same protein. Okay, and then this is the last thing, and then we'll be done for today. Oh, actually, two more things, sorry, and then we'll be done. So you need to know what a polycistronic mRNA is, and these you find in prokaryotic cells. So polycistronic mRNA. And this is an mRNA that carries the coding sequences for multiple genes. Okay, so what you're going to have is, instead of having an mRNA molecule that codes for one protein, which is typically what you have in a eukaryotic cell, this mRNA molecule actually carries the coding sequences for multiple genes. And so your mRNA molecule is going to have multiple start codons, because each protein has its own start codon. And these are common, again, in prokaryotes, but you do not find them in eukaryotes. Okay, so that's polycistronic mRNA. And then the very last thing is more of a question than presenting any new material. So the top part is a picture of an actual cell taken with an electron microscope. And then below it is just a drawing describing what's happening in the picture. So it's a picture, it's a drawing of what's taking place in the picture. So up here you have the DNA molecule, and then you have ribosomes bound to the DNA molecule making a red mRNA molecule. And then you can see that you have a ribosome bound to this mRNA molecule that's not done being made. And it'll start making the protein and translating that mRNA molecule before it's done being made. So then the question, the purpose of the slide, is this a picture of a bacterial cell or a eukaryotic cell, and why? No. No, we see, so I did forget to mention this, that you can't tell the shape of the DNA because you don't see the whole DNA in that picture. So you don't know the shape, but you can tell based on what's going on. So in what type of organism could translation happen while transcription is taking place? Right, bacteria. That's one reason. What's the other reason that this couldn't happen in a eukaryotic cell? So one reason it couldn't happen is because in a eukaryotic cell you'd have to splice that RNA molecule and process it before you translate it. So you would have to wait for it to be finished being made. What's the other reason that? What's in different areas? Yeah, you're right.
Transcription and translation
Right, and so in a eukaryotic cell, the ribosomes are all in the cytoplasm or cytosol, and transcriptions happen in the nucleus. So even if you didn't have to have RNA processing take place, the ribosomes aren't going to have access to the mRNA molecule until it's been exported out of the nucleus into the cytosol. So the short answer is that this has to be a bacterial cell because you have translation occurring before transcription is done, and that cannot happen in a eukaryotic cell. Okay, do you guys have any questions? Okay, so we'll stop there for today.